Okay, so now we are starting in a new area. We will be doing protein metabolism. And so just as a general overview, protein metabolism is going to be tied very closely to nitrogen metabolism. And you have to deal with nitrogen because if you generate ammonia, NH3, this is very toxic. And it does damage to cells, which ultimately does damage to systems and organs, etc. So with respect to proteins, we do have dietary proteins, so we will talk about digestion, similar to how we talked about carbohydrate and lipid digestion. Digestion of dietary proteins, you can see there is no salivary digestion. It starts in the stomach and continues in the intestine. And then you are going to get amino acids that will be picked up and released into the blood. These amino acids can, of course, form proteins inside your body, which are necessary. Proteins are things like your enzymes. Your amino acids are going to be containing nitrogen. And then so nitrogen, if your amino acids are used for energy, needs to be dealt with, and they're mostly going to be added to a molecule called urea or other excretory products, and then eventually everything will go out through excretion through the urine. So we have essential amino acids and non-essential amino acids. So the non-essential amino acids are, of course, synthesized in your body, which means we have enzymes and mechanisms to make these non-essential amino acids. The essential amino acids, we do not have enzymes to make them. There are some conditionally essential, and so we have cysteine, for example, because cysteine comes from methionine. And so if you have a deficiency of methionine, then cysteine, of course, is going to become essential. We also have some essentials based on growth. So we have arginine for growth. We also have tyrosine because tyrosine is going to come from phenylalanine. And so again, if you have a deficiency of phenylalanine, you are going to have a deficiency, or you're going to have to make, or tyrosine will have to become an essential. So we do have our main excretory products with respect to nitrogen. We have urea. We also have ammonium ion. So urea is going to be about 12 to 20 grams per day. You also have your ammonium ion, which is not toxic. Ammonia is toxic. We also have creatine phosphate, which is reserved energy, and you're going to release or break down your nitrogen to form creatine phosphate. We also have a breakdown of purines. So we'll talk about purines and pyrimidine metabolism in a future lecture, but breakdown of purines are also going to release nitrogen. So when we talk about amino acids, there are some basic categories. Glucogenic is basically amino acids that can degrade to pyruvate and become glucose precursors. And so the carbons in these amino acids can be used to make glucose. And then you have ketogenic amino acids. And there are two ketogenic amino acids. They are lysine and they are leucine. And what it means to be ketogenic is that these amino acids are going to degrade to acetyl-CoA. And as you know, acetyl-CoA carbons are not used to make glucose, or acetoacetate. So they can become fatty acids or ketone bodies. And there are only two of them that are ketogenic, so it's very easy to remember: lysine, leucine, ketogenic. Now there are some that are going to be both glucogenic and ketogenic. And so this is going to be isoleucine, phenylalanine, we have threonine, tryptophan, and tyrosine. And then for glucogenic, it's going to be all others. So if you remember who's completely ketogenic, lysine, leucine, and then those that are both gluco and keto, then everything else is considered glucogenic. So this is just an overview of nitrogen metabolism. So your nitrogen-containing compounds also have carbon skeletons that can be used for other pathways. But in order to use those carbon skeletons in other pathways, you have to remove your nitrogen. So removal and so we kind of saw that with alanine when we were making glucose through gluconeogenesis. Alanine went to pyruvate to become your glucose. So your fats, your glucose, the fatty acids and amino acids all kind of converge. And so, of course, you have glucose, which can be in the blood. You can take glucose up. Liver cells and some other cells can release glucose. Glucose can become glucose 6-phosphate to go to glycogen stores. Glucose 6-phosphate can become pyruvate. Pyruvate can become acetyl-CoA. This can generate ketone bodies. You also have pyruvate that can enter and become part of your TCA cycle. You have citrate that can become fatty acids. You have your keto acids, which can, through transamination reactions, become different amino acids. Amino acids, of course, can be important in many different compounds, and they're all listed here. You have your purines and pyrimidines. You have your creatine phosphate. You have your glutathione, which has glutamine. Heme is going to be made from your proteins and your amino acids. You have serotonin, melatonin, norepinephrine, epinephrine, etc. So there's a lot of things that are going to be made in that. Now, anytime you have a deamination reaction, you're going to release a nitrogen. The nitrogen has to be packaged up, and nitrogen gets packaged up into urea, and then urea gets excreted. So during fasting, your proteins, the carbon skeletons from your proteins, can be converted back to ketone bodies, or they can be converted back to glucose. So let's talk first about digestion. And so again, you're going to take up proteins in foods. Your proteins need to be digested by proteases. The proteases are zymogens, which are pre-enzymes. And so zymogens have to be cleaved in order to be activated. Some of the zymogens are going to be activated in your gut through just simple pH. Others are going to have to be activated by other enzymes. So you're going to have pepsin from the stomach. This is going to, in an acidic environment, denature proteins. So we have our pepsin. And then we're going to have our pancreatic enzymes. And of course, this is this is low pH, and this is a higher pH. And so in this, you have trypsin, which is going to be a central enzyme for your pancreas. There are others that we will talk about. And then in general, your amino acids are going to get picked up, and your amino acids will be transported across your intestinal cell membranes, and these are going to be through receptors with sodium and hydrogen. So they're co-receptors. And then you're going to absorb the amino acids. So for this part of the lecture, we're going to talk about the process of digesting your proteins and the enzymes involved in that. We're going to talk about how these zymogens get activated to actually digest the proteins and the specificity of these peptidases. We also are going to talk about protein absorption. So digestion is going to begin in the stomach with pepsin. Oops. And then you have other enzymes, the two principal pancreatic enzymes, they're not the only two, of course, are trypsin and chymotrypsin. And then you have additional ones. You have your Carboxy Peptidases, and elastin. Okay, so with respect to pepsin, pepsin is secreted by your gastric chief cells. And it's going to be released as pepsinogen. So this is a zymogen. This is a pre-enzyme. Now, your parietal cells of your gut are going to secrete hydrochloric acid. And so this is going to lead to a lower pH. And this low pH is what activates pepsin. Oops. Let's see. Sorry. So the low pH of your stomach will activate pepsin from pepsinogen. So parietal cells are going to be stimulated by your gastrin. So gastrin is through the endocrine system. So parietal cells are stimulated. And so gastrin is going to tell the parietal cells to release hydrochloric acid. Hydrochloric acid is going to drive down the pH of your stomach, and the pH is going to cleave your pepsinogen and convert it to pepsin. Now, other things that can trigger your parietal cells to become stimulated are acetylcholine from neural, can also stimulate your parietal cells, and histamine. Can also do the same thing. And so you have your endocrine system through gastrin, so this is endocrine. And then you have your neural acetylcholine. And then you have your paracrine from histamine. Gastrin is present in the G cells, and this is going to be in your gastric atrium as well as your duodenum, and it's a peptide hormone. It causes gastric motility and the growth and secretion of hydrochloric acids in the stomach. So gastrin is made and released by your G cells. Okay, and then we have trypsin. Trypsin is going to be a proteolytic enzyme. It's produced by the pancreas as trypsinogen. Right? And then this is made by, of course, the acinar cells of the pancreas. And the gastrin is going to stimulate the acinar cells to release these digestive enzymes. So we have gastrin again, which triggers the release of the enzymes from the pancreas. And then we have chymotrypsin, and chymotrypsin is also a digestive enzyme and has a serine group, the active site. So this is called a serine protease, which the serine is in the active site. So proteases are oftentimes named for what is actually in their active site, not for their substrate. And so a serine protease means you have a serine active site. Cysteine protease means you have a cysteine in the active site. And so that's how they are named. These are going to cleave hydrophobic and aromatic amino acids. Now just keep in mind that with respect to, this is a good place to introduce serine proteases because we do have what's called serine protease inhibitors. Serine protease inhibitors are given to people who have the high incidence of blood clotting because a lot of your enzymes in blood clotting are serine proteases. So even though your digestive enzymes are also serine proteases, those are not typically the target for serine protease inhibitors. And most of your serine proteases that are involved in digestion are in your gut. And so they're not in the blood where your serine protease inhibitors are trying to block clotting, for example. So just keep that in mind with respect to your proteases. The name that they're called is because that's what's in their actual active site. Now we have our carboxy peptidases. And carboxy peptidases are going to release C-terminal amino acids from peptides. Okay, and so they're going to hydrolyze these peptides at the end of the protein, and they're going to be classified by their active site again, similar to our serine proteases. So if they're metallo, that means they use a metal in active sites. And then they can also be serine proteases or cysteine proteases. So it just depends on what is actually in their active sites. Let me get rid of elastin for a moment. For carboxy peptidases, we have two major types. We have the carboxy peptidase A and B. There's also an aminopeptidase. Let's do A and B. So you have carboxy peptidase A, which is going to prefer aromatic or branch-chain amino acids. And then you have carboxy peptidase B. And peptidase B is going to like positive charges or basic amino acids. So arginine, lysine, for example. We also have our aminopeptidases. And aminopeptidases are going to cleave peptide bonds at the end terminus of the protein. So again, these are going to be secreted in the inactive form by your pancreas, and they're going to be procarboxy peptidases. And most carboxy peptidases, even though we're talking about them being involved in digestion, most of them in your body are actually involved in protein moderations. They are often there for post-transcriptional modifications of proteins, the things that we've talked about already up to this point. And then you also have your elastases. Let me see, I'm going to put that in here. Oops. Your elastases are going to be serine proteases again, and they are going to help break down not only proteins but fats and carbs as well. They will cleave our small hydrophobic amino acids. And those are going to be like glycine, alanine, valine. And they're also going to break down elastin, which is found in connective tissues from the foods that we eat. Okay. All right, so now we have here a picture, and this is just a picture of the digestive system. And so you have your gastric glands in the stomach lining. You have your parietal cells that are going to secrete hydrochloric acid. You have your chief cells that are secreting pepsinogen. And then you have your gastric mucosa, your G cells that are going to secrete gastrin. And so just remember that gastrin is going to release, and gastrin is going to cause mobility, and also it's going to cause your parietal cells to secrete hydrogen chloride. Hydrogen chloride is, of course, going to decrease the pH of your stomach, so you get a low pH, and with that low pH, you get your pepsinogen becoming pepsin. And then you can start digesting your proteins. So pepsin begins digestion in the stomach. And your pancreas is going to release zymogen granules, and they are going to be released into the collecting duct of the pancreas, which is going to feed into the small intestine. And so here's your pancreas. Sorry, let's go back to this one. So you have your collecting duct, you have your zymogen granules, and you have your rough ER, which is, of course, just like any other cells where the proteins are being made. And so these green things are your zymogen granules that are being released into the collecting duct. And your pancreas is also going to release bicarbonate. And this is going to increase the pH. And they're also going to release zymogen granules. And inside those granules are going to be trypsin, chymotrypsin, Enteropeptidases, etc. Now there is acute pancreatitis, which is obstruction of the secretion of these pancreatic granules into the intestine. And acute pancreatitis leads to gallstones. But you also don't get your enzymes secreted, you don't get bicarb secreted, etc. So it does a lot of damage. Now, amino acids are eventually going to be absorbed into your body through your intestinal mucosa. So your villi cells are going to absorb your amino acids. So digestion of proteins, of course, is going to start in the stomach. You have pepsin, which is very good at cleaving peptide bonds, and it does so at C-terminal sites, and it likes to attack hydrophobic and aromatic amino acids. So here's pepsin. It's kind of smushed here, but this is in your book too. So pepsin is going to go after phenylalanine, it's going to go after asparagine, it's going to go after tyrosine. So those hydrophobic and aromatics. And this is just a picture of your chief cells. So these are, of course, in your stomach, your chief cell and your parietal cells. Parietal cell gets HCl. Chief cell, of course, releases pepsinogen. Now, pepsin is going to be inhibited as this gastric juices pass from the stomach and mixes with pancreatic juices in the duodenum. And this is because of bicarbonate. So transfer so you have your stomach, into your duodenum, and here you have your pancreatic juice. And so this is very low pH. Oops, those arrows are not very good. And then this is higher pH. So you still have your secretion, emulsification of your food, plus pepsin. But now pepsin is inactivated because of the increase in pH. Now, and most of this increase is due to bicarb from the pancreas. And so you're going to now have to rely on pancreatic enzymes to continue to digest your proteins. And they're primarily going to be trypsin and chymotrypsin, which digest the proteins. And so your trypsinogen, are going to be activated by Enteropeptidases. Enteropeptidases are also called enterokinases, but they have nothing to do with catalyzing the transfer of phosphate groups. So these are actually also called enterokinase. But no transfer of the phosphate groups, okay? So that kind of name is kind of a misnomer. Your enteropeptidases are produced in your small intestine, the mucosa of the small intestine. These guys are serine proteases. So again, they have a serine in their active site. They're at the intestinal brush border, which is going to, they actually are anchored to the brush border membrane. These are going to hydrolyze peptide bonds, and it's going to activate trypsin from trypsinogen. Okay, so after you get trypsin made, trypsin can then go and activate all your other essential enzymes. Your chymotrypsin will become chymotrypsinogen, becomes chymotrypsin, proelastase, elastase, procarboxy peptidases, etc. So your trypsin is going to be very central because not only does it have a digestive role of being able to digest dietary proteins, it also has the role of activating other digestive proteases. Trypsin, chymotrypsin, and elastase are serine proteases, and these are also referred to as endopeptidases. Okay, so again, we had our pepsin, which favored our asparagine, our phenylalanine, and it's going to like the hydrophobic and aromatic amino acids. And now we have our trypsin. And our trypsin is going to be very specific. So trypsin is the most specific. So it's going to like to cleave peptide bonds in which you have our carboxyl group provided by lysine or arginine. Okay, and then we have chymotrypsin. And chymotrypsin is going to like residues containing hydrophobic or acidic amino acid. And then there's elastase. And elastase is going to cleave bonds in which you have the carboxyl group is small, as I mentioned before. And the small amino acids are things like glycine, serine, and alanine. And then we have our carboxy peptidases. And these are going to remove your amino acids from the carboxyl end. Okay, so next up we have our absorption of amino acids. So free amino acids and peptides that are 2 to 6 amino acids in length are going to be absorbed by your enterocytes, and usually this is by the proximal jejunum. Some other absorption occurs in the duodenum and ileum as well, and then the undigested protein is going to be picked up by your colon mucosal cells. Now, our transport across your epithelial cells is going to be a co-transport with sodium. And so very similar to our glucose and galactose, we have a sodium amino acid co-transporter, and it's on the brush border. It is an active transporter. It requires a sodium-potassium ATPase exchange. And then on the serosal side, you're going to have what's called a facilitated transporter. So the peptide can also be taken up. So this is an amino acid. And so if you have a peptide, this is going to be taken up instead of that by a sodium amino acid. It's going to be a hydrogen peptide transport. So amino acids are brought in by a sodium co-transport. Peptides are taken up by a hydrogen co-transport. Okay, so this is all again similar to our sugars, in which a low intracellular sodium or hydrogen concentration is going to provide the gradient for the uptake. Sodium-dependent carriers are on the luminal membrane, and they are facilitate transport carriers. and that's on the serosal side. Now, there are a number of amino acid transporters, and they fall into three very broad categories of neutral, dibasic, and acidic. They are all members of a solute carrier family. So that's abbreviated SLC. And they are expressed throughout the body. They're important for the brain and kidney especially. If peptides are taken up, they get hydrolyzed into free amino acids by the enterocyte peptidases, and then they are released as free amino acids. So if we go back up here, let me erase that just really fast. So this is going to bring in our peptide. Okay, and then our peptide is going to be converted to amino acids by peptidases in the cytoplasm. And so only amino acids are going to leave through the sodium-dependent facilitated transporter. Okay, most amino acids are going to be transported by more than one transport system. And in cases of starvation, your intestinal epithelial cells are going to take up amino acids and use them for energy. And so amino acid transport across that serosal membrane is going to be bidirectional. So if we go back here, this is bidirectional uptake or release. Amino acids are going to then enter the blood. And in the blood, they get transported across cell membranes in tissues, and this is primarily by a sodium-dependent co-transporter. Some of them have facilitated transporters as well. Let me get rid of this. This is the major. And this is the minor route, okay? This is going to be different from glucose, of course, in which sodium transport is in the intestinal and renal epithelial cells, but facilitated transport by those GLUT receptors in other tissue types. Whereas with your proteins, you still have the sodium-dependent co-transporters throughout your tissues. Okay, so with respect to protein turnover and replenishment, so you have an amino acid pool within cells, and the amino acid pool is going to be both from pickup of dietary amino acids and from the degradation of proteins within the cell. So intracellular comes from, So the degradation of proteins is going to occur in every cell. So with respect to proteins, just keep in mind that our proteins are going to be made in our cells. Some of them are going to have a very short half-life. Actually, most of them have a very short half-life. And others are going to be more stable and are surviving the cell for days. Proteins are going to continuously be turned over. They're going to continuously be made and then broken down because they wear out and you have to replace them. Examples of some proteins that undergo extensive synthesis and degradation are things like hemoglobin. Your muscle proteins are constantly going to break down and build up again. Digestive enzymes, and also your cells, your gut cells that constantly are sloughed off. And so you're going to constantly have to make cells and proteins in your gastrointestinal tract. So about 1/4 of the cells lining the GI tract are lost every day. And approximately 6% of the protein that enters the digestive tract is actually excreted in the feces. The remainder is going to be recycled. Proteins also get recycled within the cell. So your synthesis of many of your enzymes is induced to some sort of demand, and also your synthesis of many enzymes can also be triggered by damage by oxidation. And so the demand can constantly change, and so you're going to have mechanisms for intracellular degradation of damaged proteins or no longer necessary proteins because the signalings have changed. And many of these are going to be digested by your lysosomes and your proteasome. So with respect to protein turnover, you have proteases. You're going to have a series of proteases. So you have your cathepsins and caspases, which are cysteine proteases. Your caspases are more specific. They like to cleave after aspartate residues. You have your metalloproteases, and these are named metallo because they are going to use metal in their active sites, and your matrix metalloproteases require zinc. You have a proteasome, which is going to be a large complex, and this is going to use ubiquitin tag proteins to digest. We again have a series of serine proteases, and these are going to be in a triad. So the active site of the serine proteases is going to be a triad with histidine and aspartic acid. And you have your calpains, which are going to depend on calcium, but they're also cysteine proteases. So your cathepsins are going to be found in your lysosome. Your caspases are associated with apoptosis. Your matrix metalloproteases are going to be important in the extracellular matrix. And then your proteasomes are key for turning over protein and recycling amino acids within the cells. Serine proteases are those digestive proteins we just talked about, as well as blood clotting proteins. And calpains have a variety of roles within the body. So let's just talk a little bit about protein ubiquination. So ubiquinin is actually a protein. It's 76 amino acid long protein, and it's going to attach to other proteins. And the attachment of ubiquitin to other proteins usually is a signal for degradation, and it's a process called ubiquination. Ubiquination is considered a post-transcriptional translational modification. And it was originally identified as being what is necessary to target a protein to the proteasome. The proteasome is a complex that is in the cytosol, and it has a sedimentation coefficient of 26x S. It has what is referred to as a barrel-shaped core and regulatory complexes, and its whole purpose is to degrade proteins in an ATP-dependent manner. Now, an alternative to the proteasome complex is something called autophagy, or self-eating. Okay, so ubiquination, you have the process of adding ubiquitin, and it's two steps. You have first ubiquitation, and you can have polyubiquitation, means that you have multiple ubiquitin proteins attached to the same protein that's being targeted. And then you have entry into the actual proteasome complex, and then finally digestion of the actual protein. So in order to be ubiquinated, you have to prepare the ubiquitin proteins. Ubiquitin proteins are always going to be cleaved at the C terminus. And it's via an exposed diglycine motif. Now, the ubiquitin enzymes are ubiquitin C terminal hydrolases. Now, one thing to keep in mind with ubiquitin is that it was once thought that once a protein is tagged, it's always going to be degraded, but that's not the case. There are some deubiquinating enzymes. Oops. And these do what they say. They take ubiquitin off of a protein. And an example is BRCA1. And so what is now thought about ubiquination, instead of protein being ubiquitin has to be degraded, it's more thought of another form of regulation. Similarly to phosphorylation, dephosphorylation, a quick way to regulate enzyme activities. By coating certain proteins in ubiquitin during a certain period of time and then being able to deubiquinate that same protein in order to cause it to be activated is thought to be a mechanism of control. So you have basically 3 enzymes that are required to attach to the C-terminal glycine of ubiquitin to a lysine residue on a targeted protein. And the 3 proteins are E1, E2, and E3. E1 is simply ubiquitin activating enzyme. It's going to activate ubiquitin, and it is ATP dependent. C2 is going to be a conjugating enzyme, and this is where you're going to get transfer of actual ubiquitin to whatever protein that is being targeted. E3 is going to be a ligase, and this is going to be the process of attachment of the ubiquitin to the protein through covalent attachment. Ubiquitin is usually attached to the epsilon amino group of an internal lysine residue. And a protein can have many ubiquitin attached to it. However, as I said before, protein tagged with ubiquitin may not be degraded, and so these deubiquinating enzymes are called isopeptidases, and they are important in removing ubiquitin and also recycling ubiquitin. So what is the process of ubiquination? Well, first you have your E1 protein. And you have your ubiquitin is here. And ubiquitin is going to come in. This requires ATP. And you're going to release 2 inorganic phosphates. So you're going to release an AMP and 2 inorganic phosphates. And then this results in your E1 with ubiquitin attached. Now, after this, you have your E2 that's going to come in. And E2 is going to come in and release E1. And so now you're going to have your E2 with your ubiquitin. And then you're going to have an E3 protein, which has sort of two docking sites, you can think. And so you're going to have E2 bind to 1 with your ubiquitin. And then you're going to have your substrate bind to another. And then you're going to have your ubiquitin transferred to that substrate. And then you have your release of E2. You still have your substrate, and your substrate might now have many ubiquitins. And then this could go to the proteasome, or it can be deubiquinated or unubiquinated. And then you get a ubiquitin plus a substrate gets released. Now your proteasome is going to degrade proteins. The ubiquination or polyubiquination, the more ubiquitin added to an actual substrate is going to have more of a chance of being degraded by the proteasome. Proteasomes are a very large complex. You're going to have ATP, which is going to be required to unfold the target. And then ubiquitin is eventually going to get released and be recycled. So you have your 20S proteasome, sort of a barrel shaped shown here. And then you have here, you have your cap protein, which is a 19S. So you have your barrel shape plus your cap protein plus your targeted protein for destruction. And this complex is 26X proteasome, and this is going to need ATP. You then have your regulatory proteins, such as PA28, that gets added. And these are going to all be active proteasome complexes. So your proteasome can only have a P28. Your proteasome can have a 26S, or your proteasome can be a 26S having the cap protein plus the PA28. All three of them are active proteasome complexes, and all three of them can degrade a peptide. Now, an alternative pathway of degradation, as I mentioned before, was autophagy. And autophagy just simply means self-eating. This is a way for cells to degrade and recycle parts of the cell that are dysfunctional. It is also a way to promote survival during stress and starvation. So when a cell doesn't have energy coming in, it has to generate its own energy. It can do so by breaking things down that aren't necessarily essential for the cell to survive. However, if autophagy continues, of course, the cell will eventually die. So the three pathways for autophagy are macroautophagy, and this is where you have a lysosome, which will engulf a portion of the cytosol. You can think of it as sort of eating little chunks of your cytosol. So whatever it engulfs, it is going to digest. You also have chaperone-mediated autophagy, and this is where you're going to get different chaperone, heat shock protein molecules that are target substrates for the lysosomes. And then you have macroautophagy, and this is what is typically thought of. This is more of a traditional autophagy. So this would be 2 and 3. Macroautophagy is where your cytoplasmic organelles get sequestered within membrane compartments and digested. So lysosomes are going to be key in the process of autophagy because they're the ones who have enzymes to degrade many of the components of the cells, from sugars to proteins to fats. And so they're in the classic case in which intercellular components are surrounded by membranes. These membranes will fuse with a lysosome. So the classic, in which you have organelles within membranes, and these are going to fuse with lysosomes. And then you get digestion, of course. So within lysosomes, you have, with respect to proteins, the Pepsin family of proteases that are going to degrade these proteins. And then the recycled amino acids are going to be released from your lysosomes. Now, with respect to lysosome protein turnover, there is the Mtor pathway. So we have our Mtor pathway. Here's our Mtor. And Mtor is going to be a protein that inhibits autophagy. So Mtor on, autophagy off. Right? So during starvation, Mtor is inactivated, so the cell can undergo self-proteolysis. So again, if Mtor is on, then you're not breaking things down. In the case when a cell needs to have reagents, you're going to need Mtor to be turned off. And so rapamycin is going to target this Mtor pathway. Right? And so you can see that in the Mtor pathway, you have an input of your insulin. So it makes sense that your insulin is going to want to turn Mtor on, because you don't want to undergo autophagy while you have plenty of energy around. And rapamycin is used for transplant rejection. Oops, sorry. Okay, and it was shown that rapamycin is going to impact the Mtor pathway. And so rapamycin actually impacts it right here. It does the opposite of insulin. So rapamycin is going to block AKT or insulin, and then it's going to block this pathway here. You're going to get inactivation of Reb and inactivation of Mtor, and then activation of autophagy. Now, why would you want to activate autophagy for transplant rejection? Well, it turns out that, of course, your immune system is what is going to reject your transplant. So if you're targeting your immune system, the cells that are active, then you're trying to kill those cells before they have a chance to reject your actual transplant.